Looks like everyone has started to join. Well, I think we can go ahead and get started. Well, hi, everyone. Thanks so much for coming today. Welcome to this joint webinar co-hosted by 10x Genomics, BioLegend, and Illumina, featuring Dr. Xuan Zhang, Research Associate, Tribes Laboratory, Cincinnati Children's Hospital Medical Center. My name is Samantha Shelton, and I am a Science and Technology Advisor here at 10x. A few housekeeping notes before we get started. Please go ahead and type your questions into the chat at any time, and we will address them after Sean's presentation. So we're going to start off with some brief introductions by myself and Roman Magayan, Product Manager, Single Cell Sequencing Proteogenomics at BioLegend. So I'll go ahead and get us started. And I'm really excited to be here today to talk to you about some of the recent updates that we've had to our portfolio before we begin, and how they can also be applied to proteogenomics as well. So at 10x Genomics, our goal is to provide researchers with the tools that they need to unravel the biological heterogeneity of their most precious samples. So we all know that biology is very complex, and so being able to look at it with a high degree of resolution is very important. And we do this across our three product platforms: our Chromium single cell platform, which allows you to look at multiple modalities from a single cell suspension; our Visium spatial platform, which allows you to get that whole transcriptome readout while also maintaining the spatial architecture and tissue context as well; and then our Xenium in situ platform, which allows you to again, get a spatial look at your tissue, but from a more targeted perspective. So a panel of 500 to 5,000 genes, and with the highest possible resolution. So actually down to subcellular level resolution with Xenium. And so I'll start by talking about some of the updates that we have across these three platforms, starting with our Chromium single cell assays. So our Chromium single cell platform kind of has two different flavors of assays now. So we have assays that are reverse transcription based, so they use a poly-A based capture method. And these are really critical for a number of different types of experiments. So these allow you to capture things like isoforms and SNPs. And also, of course, because they're poly-A based, they're species agnostic. Additionally, these assays have very low cell input requirements. You can go down to even like 600 cells or so. And you would be able to capture good data from that. On the other hand, we have our Chromium single cell gene expression flex assay, which works a little bit differently. So this is a probe-based assay rather than reverse transcription based. And there's a number of benefits that conveys. So this allows for fixation, which allows for easy sample batching. It also has a built-in multiplexing feature, so you can really reduce the cost per sample down to just a few $100s per sample. And furthermore, right now, this is the only way to run single cell FFPE samples. So what that means is that you can take an FFPE tissue, you can dissociate it into a single nuclei suspension, and you can actually run that with our Chromium single cell gene expression flex assay, which just opens the door to thousands more samples that were previously unavailable. And so I want to start by covering some of the updates on our reverse transcription-based assays, which really are primarily centered around the launch of our newest platform, which is the GemX platform. And so GemX is essentially like the latest update to our reverse transcription-based assays, and it conveys a number of benefits as well. So one of the biggest updates that we have is that it has a dramatically increased sensitivity. So here you can see, looking at all these different sample types, we see dramatic increases in sensitivity. And in particular, if you look at the adult mouse brain nuclei, you can see that compared to our previous versions of our single cell 3', we're getting almost double the number of genes per cell. So this is a really dramatic increase in sensitivity with our latest update. Additionally, GemX now has the ability to capture up to 80% of your cells. So previously, NextGem, the previous assay, captured up to 65%. Still quite high, but being able to capture more cells is really important particularly for those samples that have very few cells, such as tissue biopsies or something where you're doing cell enrichment and you only have a really small number of cells. Being able to capture as many of those as possible is critical for getting the highest quality data. And so we're really excited about the increase in capture efficiency as well. Furthermore, we've also seen enhanced ability to capture even challenging cell types such as neutrophils. So for those of you who don't know, neutrophils are very low RNA containing cells, and they do tend to be fragile and die quickly. And so, with our 3' V3 version, we were still able to capture neutrophils. So you can see here that in purple, this is the group of neutrophils. But with the upgrade in chemistry and this ability to detect more RNA with higher sensitivity, you can see that we're significantly increasing, really almost doubling the number of neutrophils that we're able to capture using our 3' V4 Genomics assay. And looking here, you can see that this isn't impacted by the total number of cells loaded, like across all of these other cell types. You can see we're still recovering a similar proportion, but it's really with these challenging cells that we see the significant increase in recovery. So Genomics, as I said, has a number of benefits. I covered just a few here, but this assay is now available and researchers are using it already all over the world. And it's compatible with our X and IX series of instrumentation. So if this is something that you'd be interested in, please let us know and we can help you design your experiment. Next, I want to cover some updates on the probe-based version of our single-cell assays, which is our Chromium Flex assays. So, as I mentioned, this assay has a number of benefits, including this ability to enable sample fixation, so you can really streamline your workflow. FFPE compatibility. So we've actually even seen some customer publications, which you see here, where they're able to generate these nice single-cell suspensions from FFPE tissue blocks. And finally, of course, as I mentioned, there's absolutely massive scale at reduced cost per sample. So you can see here we have millions of cells which you can run in a single experiment using Flex. And Flex is also compatible with multi-omic characterization as well. So you can see here that we have one sample, which we split amongst 16 different barcodes. And that's how we do our multiplexing, is using barcodes. And you can see from this that we were able to capture this really nice gene expression data, but we're also able to capture this really highly multiplex protein data as well. So this allows us to better refine the cell substates, like looking at these different T cell populations, for example. And so it's really nice that this is available, not just with Flex, but also compatible with the multiplexing aspect of this assay. We are looking at expanding Flex capabilities further this year. So we actually just recently launched the ability to do intracellular protein profiling. So we're really excited about this. So this is a new functionality that is enabled on Flex. CRISPR screening. This is another new functionality that we've started to enable. So here you can see 7,000 guides detected by the Flex assay. And whole blood fixation. So this is an industry first that allows you to fix whole blood and then deplete the red blood cells from it. So this can, again, really streamline your workflow quite a bit. So yeah, all of these updates are being released this year. And so again, if you have any questions, please let us know. Okay, and then on the spatial side of things, I just want to cover a few quick updates. So starting with our Visium spatial platform, really the big update that everyone is talking about when it comes to Visium is the launch of our HD platform. And so here you can see, we have this human FFPE colorectal cancer sample, which we profiled with standard Visium. And you can see that you do get, really nice spatial data using this assay. But you can see that when we compare it to the type of data that we get using Visium, just the level with which we're able to resolve these really small tissue structures using Visium HD is really, really amazing. So we're super excited about this, and people have also started to use this already. So again, let us know if this is something you'd be interested in. And finally, a brief update on our Xenium in situ platform. So the big update on Xenium is that we've recently launched our 5,000 plex panel. So this is a pretty big update over our previous panel size. So previous panels, which are still available, were around 500 genes per panel. We've now upgraded this to a 5,000 plex panel. So we're really excited about this. It really allows you to just better profile the different cell types within your tissue. So here you can see we're looking at this lung adenocarcinoma sample. We have all these different epithelial cells, and because we have so many genes, we can more easily identify between this tumor epithelial cell type 1 and tumor epithelial cell type 2. And it also allows us to, differentiate between different immune cells as well. So we can more clearly differentiate between, cytotoxic T cells and regulatory T cells, for example. So again, very excited about this. These are, some of the updates that we have announced for this year, and we look forward to seeing what will be coming next. And with that, I will go ahead and turn it over to Roman.
Thanks for the introduction, Samantha. Good morning, everyone. Can you hear me okay? See everything okay?
Yes.
Okay, perfect. Good morning, everyone. My name is Roman Magallon. I'm one of the product managers covering the proteogenomics portfolio at BioLegend. And today I just quickly wanted to review some of our multi-omic product offerings and solutions. So currently we offer 4 formats that integrate with the 10x genomics applications, each with over 500 catalog products. That's with the exception of our newest format, TotalSeq BN, which we're currently working on expanding. TotalSeq A antibodies were the first conjugates we developed for CITE-seq. They contain a poly A tail that mimics mRNA and can be captured by platforms that utilize poly DT capture sequences. Our TotalSeq B, C, and BN formats were developed in collaboration with 10x Genomics. TotalSeq B is compatible with 10x's 3' gene expression assay and gene expression flex assay with feature barcoding for protein. They contain capture sequence 1, which is complementary to the capture sequence 1 found on the 10x gel beads. TotalSeq C is designed to work with 10x's 5' immune profiling kits and are also compatible with 10x's gene expression flex assay that utilize the barcode oligo capture for protein detection. And then finally, we have TotalSeq BN that can be used with 10x's Visium spatial gene expression solution for FFPE tissue sections. And similar to TotalSeq B, it contains a complementary oligo sequence to the capture sequence 1 found on the oligo lawn of the Visium slides. Now taking a closer look at our pre-optimized antibody cocktails, which provide excellent value to customers. These cocktails are provided in single-use tubes, are pre-titrated and optimized using an NGS readout. This eliminates the need to manually combine individual antibodies, which help reduce inconsistencies with pipetting or handling of multiple vials and samples. We currently offer 4 different types of cocktails, our most popular being the universal cocktails, which contain up to 163 unique antibodies for the identification of principal immune lineage cells and sublineage types. They're available in formats of A, B, and C for both human and mouse cells. On the other side of the spectrum, we have our smallest cocktail, the human TB&K cocktail, which contains 9 antibodies and is used for the identification of principal lineage cells. These cocktails are great for customers that are just getting into the single-cell research space and would like to explore the use of antibodies for the detection of protein. And then finally, we have our newest cocktail, which is the TotalSeq B mouse myeloid cocktail that was optimized for the identification of myeloid cells. It is currently only offered in TotalSeq B, but there are plans to expand it into some of the other formats. Our TotalSeq product offerings can be broken down into 4 main categories. The first being our primary single-cell antibodies that feature over 500 unique clones or greater than 1,800 products across formats in human and mouse specificities. These individual antibodies can be used to create your own antibody pools or used as spike-ins to additional antibodies into our existing cocktails. The pre-optimized antibody cocktails, which I covered in the previous slide. And then we have our popular TotalSeq hashtags that can be used to sample multiplex. We have over 1,500 hashtag products that offer 24 hashtags in each of the formats for both human and mouse cells. We also offer nuclear hashtags for multiplexing nuclei. And last are our TotalSeq secondary reagents, which include products such as our streptavidin oligoconjugates, our anti-tagged products such as our antibiotin and anti-fluorophore conjugates. And as you'll hear today, BioLegend offers TotalSeq custom cocktail solutions, which really gives you the ability to design a panel that's suited to your needs. These cocktails are typically suited for scientists that have a pretty clear idea of the content they want in a panel. And usually, it's based on existing data. We often suggest a custom panel when TotalSeq catalog cocktails are not the best solution, or if customers are working on novel applications. For these custom cocktails, there is a 15-plex 50 reaction minimum, and the lead times typically range from 6 weeks to 16 weeks, depending on the project. And next, I just wanted to make you all aware of BioLegend's new validated protocol for the detection of both surface and cytoplasmic intercellular proteins that is compatible with 10x's gene expression flex assay using our TotalSeq B antibodies. This protocol allows for simultaneous detection of hundreds of cell surface proteins and cytoplasmic intercellular proteins with gene expression when it's paired with 10x's gene expression flex assay for single-plex samples with feature barcode technology. The ability to look at all three of these modalities, surface, intercellular, and gene expression, gives customers an even greater means of characterizing cells. It enables the identification of functional molecules and can provide insight into signaling pathways. As of today, we currently offer 30 cytoplasmic TotalSeq B antibodies that customers can use to assess intercellular expression, and we will continue to release more markers in the near future. To learn more about the intercellular protocol and available antibodies, be sure to check out our new intercellular webpage. And finally, what's on the horizon, what we're working on, and what's next for TotalSeq. First, we're in the process of developing our new generation of pre-optimized immune cocktails, 3 different cocktails of varying sizes ranging from 100 to 300 antibodies. This new iteration of cocktails builds on what we have learned based on customer feedback and review of the literature to expand on important markers and dimensionality. Each cocktail will contain critical markers for the classification of multiple lineages of immune cells and antibodies that are specific for cancers and cancer therapeutics, as well as markers spanning different tissue types. The first of these cocktails will hopefully be launched within the next few months, and the rest will follow into 2025. On the intercellular front, we have plans to expand the TotalSeq C format antibodies. We are also working on protocols for the detection of nuclear, transcriptomic, and phosphoproteins. We're hoping to launch the TotalSeq C intercellular antibodies by the end of Q3 and the protocols hopefully before the end of the year. And thank you. Yeah, I'll give it back to Samantha. And I know I went through that pretty quickly, so if you have any questions, feel free to reach out. We'll be sure to share our contact information. Thank you.
Samantha, you're on mute.
Thank you. Thank you, Roman. So with that, I'll go ahead and introduce our speaker today, Dr. Xuan Zhang. So Dr. Zhang is a rising researcher from the Grimes Lab at Cincinnati Children's Hospital Medical Center. He studies hematopoiesis and the pathogenesis of hematologic malignancies, such as myelodysplastic syndrome and AML, using cutting-edge single-cell multiomics techniques, including both NGS and long-read sequencing. His research focuses on uncovering the determinants of hematopoietic cell lineage specification and disease progression by simultaneously profiling the epigenome, transcriptome, spliceosome, and cell surface proteome in single cells. The title of his presentation today is Defining the Bone Marrow Atlas with CITE-seq Workflow and High-Dimensional Flow Cytometry. And with that, I'll go ahead and turn it over to Xuan.
Yep. Thanks for the introduction. I cannot wait to share my screens and tell you all about our recent publication and this story, where we use CITE-seq and the high-dimensional flow to build a human bone marrow atlas focusing on the progenitors and also on the stem cells. So as we know, single-cell RNA-seq has really redefined hematopoiesis, changing it from a classical model where every single differentiation is stepwise from long-term HSC all the way to mature cell types, now to a more continuous model where you can find the transitional cell types or cell states in between. For example, back in 2016, we described a rare metastable intermediate cell state that can give rise to monocytes and granulocytic precursors as regulated by the level of GFI1 and the IRF8. However, there are still some gap in knowledge in the field. For example, we know all the facts we know are based on sorted and functionally validated cell type or cell states by flow. And all the data we have from single RNA-seq is just a hypothesis. So what really can be used to bridge it together is antibody. So, but how to bridge them? We have to use some multi-omic technologies. So some of them have already been established, for example, CITE-seq and also Mission Bio's platform, you can combine antibody with DNA sequencing. So a brief introduction of CITE-seq. So CITE-seq is developed in New York Genome Center, full name is Cellular Indexing of Transcriptome and Epitopes. So similarly to flow cytometry, you use the antibody, but this time the antibody is not conjugated to a fluorochrome, but instead with the antibody barcode that can be later on detected by sequencing. So the ADT, which I'll talk about more today, is short for antibody-derived tag. So it's basically just an antibody that has a DNA barcode on it. Also, something else we used and also mentioned by BioLegend is the cell hashing. So this typically use a hashtag oligo, also short for HTO, that binds universally to the cell types or cell states you are studying. So for example, in human is CD298, in mouse is more of CD45. So it binds to all the cell types in there, but you can use a unique HTO to label every single cell population you're staining, and later on they can all be demultiplexed. So even with powerful tools like CITE-seq, there are still many gaps in the field, like a lot of questions that can be potentially resolved. For example, right now the existing markers are not sufficient enough to really resolve and isolate hematopoietic stem and progenitor cells. And also, the commonly used ADT panels, like the universal panels, are very limited just based on the immunophenotypes and usually not context relevant. For example, titrated on PBMC, you use it on like bone marrow. A lot of the markers will occupy a lot of sequencing space like CD71 because they are not optimized. So to fully explore the power of CITE-seq in getting to novel discoveries, we designed a top-to-bottom screening workflow. And through a rigorous titration, we try to identify novel or repurposed known markers for isolating those cell states or characterizing those cell states. So here, the first task is how do we really determine useful antibodies for human marrow and their best working concentration? So this is the overall study design. So we collect fresh bone marrow from diverse cohort of donors spanning male, female, and also black and white. We get their fresh marrow overnight shipped, nothing frozen, never frozen. And then we use MAX to enrich for CD34 high cells, which are the stem and progenitors. We did a co-elution with 271. This covers nicely the more mature cell progenitors and also the stromal cells. And finally, all the cells in the marrow at the bone marrow and total nucleated cells. Then we stain these three different cell populations with a diluted concentrations of antibody cocktail that contains 266 targeting antibody and 9 isotype controls, and then label them with HTO. So in the end, for every single cell, we have the HTO that labels concentration and can be later demultiplexed. We have the RNA, the transcriptome, and we also have the surface antibody expression profile as detected by ADT. We constructed a atlas using RNA, using the transcriptome data alone for like over 300K high quality single cells. And we use this RNA transcriptome atlas to determine the performance of all those ADTs at different concentration. So a bit more details in the sample processing. So basically, all the cells were stained with, and the cells were aliquoted and stained with different concentrations of ADT cocktail, including a prototype PBMC universal cocktail that is already discontinued. And they are labeled with each individual unique hashtags, and they are washed on the Curiox lamina wash platform at 25 cycles, flow rate of 10. And then the cells after washing were put together and loaded onto 10x 3' HT version 3.1. So every single well were super loaded. We loaded like up to 130,000 cells per well. Similarly, for the TNCs, which is not really what we are interested in, the bone marrow nucleated cells. So those we typically just load in a regular 3' version 3.1. So the cell number is much lower compared to the stem and progenitors in our atlas. Also a bit more details in the library sequencing requirements. So we typically target our transcriptome library at a depth of 50,000 read pairs per cell, and sequence on a larger sequencing platform like NovaSeq 6000 S4, or on the newer one, the X Plus, the 10B version. For the ADTs and HTOs, we generally sequence on NovaSeq 6000. For the ADTs, we usually target 12,000 read pairs per cell. And for the hashtag, you need much lower reading depths because it's basically a binary result whether it got stained by this antibody on by the HTO or not. And notably, if you want to sequence them all together, it better be sequenced at pair end 100. And you should avoid the sequencing ADT or HTO libraries at longer setup, like pair end of 150 on their own, because their size is relatively small, only like 200 BP. So it will read through adapter and even read into the matrix. But you can like mix it with other types of libraries and it will work just fine. So again, just like showing you what successful hashing looks like. On the left-hand side is actually a sample included in this study. We have a great nice separation of cells that truly positively stained for this hashtag versus those are like really didn't get stained. And looking at the data matrix, you can see it really stands out to covering like over 60 or 70% of the total UMI. It's all from this hashtag one. On the right-hand side is a failed hashing experiment. You can see it does not like really distinguish well. These might be the positive cells, but they are so close to the negative ones. And looking at the data matrix, everything is pretty much like evenly distributed. So there is no way you can demultiplex these samples later because the hashing failed. And to get a good hashing, you need not only a good wash, but also need to have fresh hashtag antibodies. So back to the topic, we have a lot of RNA collected from over 300 single cells. So the next step is to construct a good transcriptome reference. And to do that, we use SC-triangulate, which is a decision level integration that can be used for uni and multimodal dataset developed by Frank Lee in Nathan's lab. So here we take into consideration of pretty much all the previous atlas and also unsupervised clustering. And in a game theory setting, we determine which one really contribute most to the stability of that cell cluster. And the clear winner will carry its annotation to the final atlas. So here is a view of the UMAP of RNA-only atlas, and this is already an expanded repertoire of hematopoietic cell states covering over 80 single cell states. And you can nicely see that over here, the CD34 high cells, which covers most the early and stem and progenitor cells, and also like CD34 to 71 coelution gets a lot of stromal cells over here, and also nicely all these precursors and the later stage progenitors. And also the total nucleated cells, we got all these lymphoid tiny cell populations. So everything worked out as anticipated. And all these clusters have very specific cell markers as shown here. But this is just a beginning, just a foundation for us to evaluate the ADT performance. So how do we evaluate ADT performance in a complex setting? There's really no consensus. So we have to do like something like, we have to do something that is based on good rationale, but there could be like other more like reasonable or better ways to do it. So I asked Kyle to do a classification model by XGBoost that ranks all these ADTs at different concentration based on their ability to separate all these transcriptome-defined clusters. So for those constantly not performing well, meaning in the bottom 50% in all concentrations, we tend to exclude them. And for the ones that seem to be working, we then look at its dynamic range change as you titrate down the concentration. So some of them are dose dependent and some of them are not dose dependent. So for the dose dependent, for example, here in the panel B and C is showing CD38. You can see across all these donors and in all these different cell capture of populations, you can see a nice titrating down of the dynamic range as you decrease the concentration from 4x all the way to 0.25x. And it's very consistent like across all the data. And furthermore, we also check the cell, we also check the dynamic range in a cell cluster specific way. For example, CD38 in plasma cells is very highly expressed. In the CLP, the common lymphoid progenitors, the expression level is okay, but you can see the dynamic range is still dynamic range change is still kept. But in the HSC, the hematopoietic stem cells, we all know that CD38 shouldn't be expressed over there. And there's like very minimal background staining of CD38. So following the concept of staining index in flow cytometry, basically we look for the minimal concentration that can retain 70 to 80% of the overall dynamic range. In this case, it would be the 2x. So we determined 2x concentration would be working the best for CD38, and then that's how we choose their like best working concentration. Is certainly not the most optimal. May not be the most optimal because we only test the 4x to 0.25x, but it's already a pretty good working concentration. We also meanwhile visually check the expression and for the background staining of all these antibodies on UMAP and on heat map. We rescued some well-established surface marker by consulting with BioLegend. So after this very complex molecular titration, we finally ended up with a titrated cocktail consisting of 132 primary antibodies. And we asked BioLegend to make it into a lyophilized product and then later on tested on 4 additional donors. So here are the performance comparison, titration versus titrated. Everything on the left-hand side is the titration data, and everything on the right-hand side is the titrated data. So all these smaller columns in the titration data is one concentration, so it's very like narrow and busy data. And everything over here on the titrated data set, every single little column is one donor. So already you can appreciate the increase of dynamic range in most of the antibodies as we decrease the total number of antibodies in there. But noticeably for antibodies that were used at a pretty low concentration in the PBMC, for example, like CD4, after we increase the concentration for the bone marrow cells, it really started to label the T cell very well. So everything here is like we increase concentration. Everything here is we decide to decrease their concentration in the final titrated cocktail. So here an example that we decrease the concentration and then it actually increases the specificity. is CD150E1. So it was not like really staining very specifically, but after we decrease its concentration a lot, it now really seems to stain for NK cells. So now with the titrated CITE-seq data from 4 additional donors, we can really use it to establish a multimodal atlas. That is our ultimate goal with this cocktail. So to do that, similarly, we use ASCII triangulate, but now the clusters are from a weighted nearest neighbor Leiden clusters at different resolution, basically resolution 1, 2, 3. So we integrate the clusters from the weighted nearest neighbor, and also we integrate with the RNA-only data. Finally, we reached to a UMAP representation of this 89 RNA plus ADT cluster. So everything is a multimodal cluster. The clustering is based on both RNA and also ADT information. And here to look at what cell population really benefits from the addition of ADT information. We check their self-classification score over here. You can see a lot of the lymphoid cell types like the B and also the T cells really gained increased classification accuracy. And also some of the later erythroblast and also cells in the erythroid lineage. So here just showing like all the clusters are based on solid specific RNA and also ADT markers. Everything here is in the supplemental table. You can all check it out. And the next thing that's very naturally occurred to us is we can do ADT to gene expression correlation analysis. For example, I like to find what novel markers can be indicative of cell fate. So for example, here, by a very simple Pearson correlation, we can see C5L2 and T-span33, so everything here in green color is ADT, actually correlate positively very much with Azure1, Elaine, all these granulocytic precursor progenitor gene or maturation genes. But they are negatively correlated with CD74, SPINK2 that marks the progenitor, meaning these 2 markers are likely something specific staining for the granulocytic maturation or commitment. Also, we look at this marker expression in at the single cell level. We align all these clusters, presuming they are like the GMPs are less mature and these are more of committed progenitors and the precursors. We can see they are very much correlated at single cell level and anti-correlated with Azure and Elaine, and anti-correlated with CD34. Similarly, we can also analyze the co-expression pattern of ADTs by themselves. For example, CD326, which has been previously found not very long ago, is a erythroid marker, but they are not sure like what's the specific stage it really stains. And also CD235A, the GPA, which stands for mature erythroblast and also red blood cells. This is a very established and commonly used marker. Their expression patterns seem to be rather mutually exclusive, where CD326 gets progressively increased in the ERPs, but turned off later on in the erythroblast that 326 really picks up. And also, we found that CD326 is somewhat anti-correlated with the expression of NMU, that is a secretion peptide. We didn't validate whether the microenvironment is in play, but these are all the directions that open up for further investigation. So now we have these hypotheses. We kind of know like what markers seem to be staining for what lineage, and the next thing is we need to validate them. To validate them, we really need to isolate the cells. So here we develop a novel isolation strategy of all those transitional cell states with paired CITE-seq and also high-dimensional flow cytometry data. And to do that, we used infinity flow. So infinity flow, let me probably describe how it was done here. So the cells were first stained with 22 antibody backbone. Assuming that 22 antibodies in there could comprehensively get all the characteristics of most of the cell populations in there. And they were split into over 100 individual wells with 1+ antibody in P channel. So everything P channel, there's no difference in terms of their signal intensity for the one additional antibody. They were incubated staining, and also like after that sampled on Aurora. So based on the similarity of the backbone antibody, that 22 antibody, the 1+ antibody co-expression pattern with the other antibody, with the other backbone antibody can be imputed back, so that in the end, one FCS file that contains 133 with one extra HLA-DR file is integrated. So that way we have one single FCS file for every single donor that you can interpret this co-staining pattern of all these antibodies in there. And this is matching the complexity of our CITE-seq data. Notably, 4 of them are also like donor matched with our CITE-seq data, so they can be directly compared for their signal distribution. Kyle in our lab developed a Python version of Infinity Flow that really enabled us to do Infinity Flow integration at such large scale. So here are some examples and the important question that can be addressed by this study. So in our paper, we provided 3 demonstrations of utility, how people can use our data sets to fuel their own discoveries. For example, there's not really any like positive marker for granulocytic commitment. We got some clue from the C5L2 and T-span. We're gonna validate them, right? And also, people are constantly facing contamination cells in their favorite flow cytometry immunophenotypic gate. For example, in this case, it's Diane Cross most loved MEP megakaryocytic erythroid progenitors. We still have some GM potential cells contaminating this gate. And also, we can identify new marker combination that could stage erythroid blasts in flow cytometry. Later on, I'll show you that. So for the first demonstration of utility, we can see that these pre-new and MNU1 cells are positive for T-span 33 C5L2, but they are negative for more mature markers like CD64, and also this marker labeled for all these monocytes population. They are high for CD117, somewhat dim and low for CD34. Then we can check all these marker expression patterns in Infinity Flow. And also based on the knowledge we got from Infinity Flow, because in Infinity Flow, you can get all these cells based on your knowledge of like already known immunophenotypes and to determine whether they make sense. So our strategy here is we gate for the CD34 dim and 117 positive cells. And then we gated out 64 and 16, which is mature marker for neutrophils. And then later on in this gate, we just gate everything based on T-span 33 and C5L2, and we can see the cells that are high for both markers seems to be CD34 low. This is the intensity map for CD34 expression. And also you can appreciate how similar and consistent Infinity Flow imputation looks like flow cytometry sorting. So it's pretty much like very similar. So the knowledge you gain from Infinity Flow can be used to guide how you set up the sorting gate in your actual flow cytometry experiment. So then we just validated by sorting all the cells. We did a morphology check and also a CFU. You can see the high cells seem to contain those granules that are present in the granulocytic precursors. And also the CFU assays tells us the high cells are pretty much like all granulocytic potential. And also they are low clonal genicity as you should expect because they are 34 lower. So next, a demonstration of utility number 2. So how we can find novel markers is actually a new marker to really purify some existing immunophenotypic gate. because the flow data and CITE-seq data share the same amount of antibodies, you can kind of simulate the flow cytometry gate in CITE-seq data. In this gate, we simulate the cells in the MEP gate, the megakaryocytic erythroid progenitor gate. This is a gate, immunophenotypic gate defined by Diane. And we can see already some markers seem to be really bifurcating this population. For example, CD133. So I was curious, like, what does it really separate apart? So interestingly, actually, the CD133 top half cells, the cells that highly express CD133 are actually like multipotent progenitors with just a little bit of MEPs, a lot of GMPs. So I suspect these cells would be the contaminating origin of those GM potential clones. Whereas the 133 bottom half are really enriched, is really enriched for MEP, MKP, and also early erythroid progenitors. So potentially this marker can be used to further purify the true MEP cells. So then you can also check the marker expression in infinity objects to see if they make sense, like CD34 expression in this case, and also CD71 expression. And also you can check the forward and the side scatter of all those cells in infinity flow. So then finally, we just validate that we sort the cells in the MEP gate based on the level of 133 expression into high, mid, and low. You can see in the donor 1 and donor 2, the 133 low cells really recapitulate the potential of MEP. That is like some unipotent to the megakaryocytic, some to the erythroid, and also you have plenty of bipotent. Similarly in donor 2, but the fraction is different in different donors. So that's why I chose to show them separately. For a demonstration of utility 3, so here, if we look at something we already have a clue in the CITE-seq data CD326 and CD235A, we can see their expression pattern is pretty interesting. So the CD326 cells are positive, and then it kind of drops the expression of CD326 a bit and become 235A positive, and eventually lost all the expression of 326 and become GPA positive. This is very consistent in the infinity flow and also in the actual flow cytometry sorting gate. So the cells in these 3 different gates also varied in their CD71 expression and also forward scatter. These are the things that people previously used to gate for different stages of erythroblast. And after sorting all the cells, we did the morphology check, and then we count the frequencies of different cells in there. We can see the 326 single positive cells are mostly enriched for pro-erythroblast. And the double positive cells contains mostly basophilic and also some polychromatic erythroblast. Whereas the CD235A positive only cells are mostly consists of the red blood cells and the more mature erythroblast stage. So this gives a very clear and granular separation of terminal erythroid poiesis. So after that, another thing we can check is because we have paired the CITE-seq and infinity flow, we can check the surface marker expression consistencies across donors and technologies. So we use the CyCombine to integrate the data from CITE-seq and also from flow. And for example, here is like, this is the donor flow data, the donor CITE-seq data. You can see their general intensity in all the cell population over there is very consistent, whereas the example over here is not. So we did both a visual check, but also a K-sampled Anderson-Darling test that to test whether the distribution of the density plots are, if segmented, if binned into smaller bins, are still consistent. So we found actually only 37 out of these many antibodies are very consistently expressed across platform and across donors. Certainly, there are more of the technology, batch effects, or noise that cannot be ruled out. But this is our observation, and definitely need more donors to validate this. So here are like what a Pearson correlation heat map of all these 37 markers for what cell population they can specifically mark. You can see like it roughly covers most of the cell states and cell types in there. So finally, this is a atlas, and we have all these antibody and also CITE-seq ADT expression. So we can use that for a tool to align malignant cells on there. In this case, it would be the acute myeloid leukemia cells. So we aligned Craig Jordan's primitive leukemic stem cells mixed monocytic leukemic stem cells and also the mixed leukemic stem cells back onto our reference. You can see clearly tells like what are mostly similar in their transcriptome to our cell states. For example, these monocytic monos, classic monos, and here would be the LMPP and multipotent progenitors, all these things. And also we constructed a azimuth that can be used to impute the ADT expression when you align data that does not have any CITE-seq modules in there. So here is a comparison of the imputation versus the actual value. You can see a lot of them are pretty consistent and also remain the differential between this monocytic and the primitive leukemic stem cells. Furthermore, we tested our titrated cocktail on three additional AML samples from Craig Jordan. So you can see like there are more markers that we can define that are differential between primitive LSC and the mixed type of leukemic stem cells. Also, this is in a cluster specific way. So these all provide clues for further diagnostic or disease progression monitoring as surface markers of interest. So I would like to acknowledge people in our lab, Annie, Kyle, and other RAs and people who participate in this project. Frank, who did a lot in the assay triangulate part, and Dr. Salamonis, our collaborators at Yale, Diane Cross Lab, Max, who did the colony formation assay, me and Everett, and also Dr. Harinder Singh and Craig Jordan for their advice and their materials. Also, I'd like to thank our collaborators in the industry, BioLegend, current and former employees, CITE-seq Monica, and Curios, who provided us a prototype, some prototype stuff, and also people at 10x for their sales and also technical support, and also Illumina in terms of all these troubleshooting with sequencer runs if it failed. And finally, here is the thing I would like to bring up. So we have a bone marrow cell atlas portal that is available to everyone. And here is like the raw data. You can find the flow integrated flow data in the flow repository. And also we provided 4 tools and the viewers. So the first thing is the azimuth viewer, where you can use it to align your data set, whether it has ADT or not, does not really matter, just align it over there. You can also do it in a by cell harmony. This is what we do mostly in-house. There are tutorials over there. And also this is a viewer for both titration and also titrated data set. You can view the marker expression in all these 89 cell states and cell types. And here is like more of easy to use viewer, I would say. So this is a gene viewer. So you can view the gene expression across all these 89 clusters in a donor specific way. So every single column over there is one donor. And over here is a surface marker viewer. So you can view all these 132 antibody expression across all these 89 donors, all these 89 clusters, and also in a donor specific way to check their donor expression variance. And also to check for your study, like if this marker stains potential cells of your interest, and if it makes sense, and further just by looking at the co-expression pattern with other surface markers, you can think of strategies to isolate them, and also use the flow cytometry data to guide it. But finally, like everything I want to say is like all the data from CITE-seq or whatever single cell RNA-seq modalities are just is just a hypothesis. So in the end, you always need to validate them independently, like flow cytometry sorting, CFU assays, or whatsoever. So just keep it in mind, it's always a hypothesis. That'll be all. And I'm ready to answer any questions. And if the Q&A session is just limited, you can reach me out yeah, with emails. All right.
So thank you, Xuan, for that excellent presentation. We do have time for Q&A, and it's perfectly fine if we go a couple minutes past the hour. We will not cut things off right at 9 p.m. Pacific time. If Raman and Sam, you're able to come back on as well. I see some questions in the box. If you do want to submit questions, please add them to the type them in the Q&A where it's pointing to the arrow on the screen. And we'll start off with one for Xuan. So are these sites, the Atlas portal, are those only available for bone marrow cells?
Yes, they are only available for human bone marrow cells with a specific enrichment or focus on the stem and progenitors. So, you know, like in hematopoietic world, there are clearly a division, people stem cells, and clearly people going on the like immune cells, like true immune cells, T cell, B cells. So we are more on this branch. Yeah.
Thanks, Sean. Now we have one for Sam. I will say, it says GEMX looks great, which we always like to hear. What are the major factors impacting cell recovery rate?
Yeah, that's a great question. So, I mean, the exact components that lead to like different performance improvements are, you know, sort of proprietary, I would say. But, you know, in general, the improvements that we see in the cell recovery rate can be attributed to the changes in the microfluidic chip that we've made. So it has like a completely different microfluidic system. And if that's something that you're interested in learning more about, you know, definitely reach out to us. We can put you in contact with our, you know, your local team. And then the increases in sensitivity, of course. So that allows us to pick up, you know, more cell types that previously would have been, you know, really challenging to pick up because of their low RNA content.
Cool. And then we've got a question that maybe for either Sam or Roman. Are any of TotalSeq B, C, or biotinylated compatible with the 10x Multiome platform?
Yeah, I can take that one. Yeah, so that's a great question. So like, you know, there isn't so Multiome uses a poly A based capture method. And so TotalSeq A is actually compatible with the Multiome assay. Yeah, it's not a fully supported part of the portfolio. So the fully supported Multiome workflow will get you gene expression and open chromatin data. But there have been a number of publications where researchers have actually used the TotalSeq A application from BioLegend, which is really amazing, to also add, you know, cell surface protein data to that too, or even intracellular protein data. So there's, you know, ways to enable that.
Okay. We actually have a few more for Xuan in here. You use the bone marrow aspirate. How well does it reflect the bone marrow tissue with its niches?
That's a very good question, because, you know, it's bone marrow aspirate basically use a large needle to puncture through the bones and only get a small aspirate. So compared to something else, so we also collect femur head from aged donor to kind of get aged somewhat atlas. It's like in development. The cell composition, I would say, might be different. So I do not know like whether you can sacrifice the human to like cut its femur open and get all the cells in there to get a better idea. But I think it's pretty much say like right now limited by the availability of the material. Bone marrow aspirate is still the way to go.
Okay. And then following up on that, amazing work, Xuan. And once again, we all agree. Is there a similar infinity flow atlas for mirroring bone marrow?
Yes, actually, we have a paper that is we have a paper right now that is being submitted possibly, so that's the leading author over there is Kyle. So he did even a better job than what I did. It will be a trimodal, combining both bulk and single cell, meaning also the epigenetics, the attack, and also all these attack related motif analysis, and the link back to all the cell types in the murine bone marrow in terms of like their transcriptome, their ADTs. So all these three modalities from attack, RNA, and ADTs are connected together by CITE-seq. Yeah. So just like staying tuned, and I hope that paper can get out soon and be published. It will also be a very good resource, but beyond the resource, there are very exciting findings in that paper. Yeah.
Yeah, we look forward to seeing that published as well. So a couple of ADT related questions following up on that. So do you have any suggestions or tips about multimodal integration of CITE-seq, RNA, and ADT? How did you do it, and why did you do it that way? And then how do you determine if ADT is signal or noise? Do you have a fixed cutoff, for example?
Yeah, that's also a very good question. I think currently when people integrate RNA and ADTs either by Surratt multimodal weighted nearest neighbor, or by something like ScanPy, or on the Python part like ScanPy, Muon, and MoFA, those kind of things. MoFA use a different background subtraction method, which I found is not so different for a good sample like ours. But it might like change a lot for other cell types. And as for comparing the antibody signal to the background, so because in every single antibody cocktail, you always have the same isotype matching isotype controls, so usually like 5 to 9. So you can compare your antibody expression pattern in UMAP and also in heat map, and also even do correlation analysis to those isotype controls. So that gave you an idea whether this antibody signal is completely BS or it is specifically standing for some cells.
And related to that, and apologies if you kind of covered this already, then should you gate for those positive and negative cells separately, as in sometimes done with flow, or is it the same threshold that you apply for all samples, like based on the ADT level?
I'm not exactly sure what kind of gate or threshold they are talking about. Or if you're talking about the specificity thing, it's just like it's like by actually boost classification models, see if they are useful for us, and also check the background if they are okay. But in terms of like positive versus negative, I mean, if you do plenty of flow, you already notice like a lot of the markers are not really a clear cut. So is in CITE-seq. So it's usually a like multimodal distribution. You may have more peaks, and also sometimes it's just a shmoo. Yeah.
And then maybe since we have a CITE-seq coming up quite a bit, some questions in there for Roman. Will the AB antibody titration considerations be taken into account for future product components? If not, is there an analysis pipeline that can be correct for such considerations that are shared with users?
Yeah, I guess this is the two-part. So the first one would be for the new cocktails that we are developing, we are tweaking some of the concentrations included. When you have a larger panel, kind of as we've seen, it becomes very important to titrate to make sure that you're not over-consuming reads or underrepresenting certain populations, because, the staining is just not there. So we are expanding on that. If it's a marker that's not included in the cocktails that we provide, then we provide guidance on how to do titrations to find the optimal concentration to include. Bioinformatically, there are also methods, kind of as we've seen today, to look to make sure that the staining is correct, but then also to figure out if that concentration needs any adjustments. But yeah, if this is something that you're working on, feel free to reach out and we can kind of guide you through the process.
And then maybe this is more of a general kind of theoretical question for the whole panel. CITE-seq gives a much higher throughput compared with flow cytometry. Given that flow cytometry is still a quote-unquote gold standard, could you compare the dynamic range of these two methods, kind of CITE-seq versus flow?
Yeah, I would like to answer that. So first, I probably need to correct this part. CITE-seq throughput is far less than flow cytometry. Flow cytometry, you can sample like at single cell level millions of cells within like 30 minutes or like like 1 hour or something. CITE-seq, we are still looking at, in this case, a maximum of 500,000 single cells high quality. So the throughput flow is much better. In here, we did compare the dynamic range, but it's really hard to tell whether this is a technical like bias because, you know, CITE-seq is measured by sequencing everything, just one count appear once, you count one, very simple. Flow cytometry is more of you have an analog signal and you determine the peak, right? And then you do a analog to digital conversion, and there's plenty of signal amplification in there. So the signal in the end, to me, I consider the flow cytometry is more kind of distorted or manipulated than sequencing. So the dynamic range scale-wise is absolutely different, but you can scale fit them into a similar scale and compare. That's what we did, and also other people did, like the CycleBind in this case. You can definitely compare their dynamic range in terms of separation, not the raw value, but also more importantly is to compare their multimodal distribution. Like, are there 2 peaks in flow? Are there 2 peaks in CITE-seq? What are the distribution, whether they are similar? That is more a critical question than dynamic range.
And then maybe we can wrap things up with a couple of related questions on cell number. One kind of maybe more for Sam, which is, you know, for initial investigations, how many cells do you recommend for NGS? And then there was another question, I think both for Samantha or Xuan, in terms of recommending overloading with 100,000 cells compared to more conservative 20,000 cell overloading.
Yeah. Yeah, I mean, when it comes to the minimum number of cells, it really depends, I guess. But like the first question is like, well, how many cells do you have? So, you know, if you're kind of working with a more limited cell population, then a lot of times what people will do is just, you know, prepare the cells, particularly if they're enriching for a rare cell population, enrich for those cells, and then basically just load that entire cell population into the 10x chip. Again, that's assuming that it's like a smaller population, less than like 20,000 cells, something along those lines. On the other hand, you know, when it gets to, if you have more cells than that, then you can start thinking about, you know, doing things like superloading or maybe like running multiple channels. And that I think sort of depends on your like the cleanliness of the sample, and then also, you know, how okay you are with the risk of like, you know, superloading. With superloading, if your sample does have some debris, that does increase the chances of there being a microfluidic failure. But if your sample is pretty clean, then, you know, the channels are still pretty robust. And so obviously, as we saw from Xuan's presentation, like he was going, you know, quite high with the number of cells loaded. So it's definitely possible to go pretty high. But, you know, for maybe those like earlier experiments where you're just getting started, I'd recommend, you know, staying within our recommended range. But then as you get more comfortable, as you start thinking about scaling up your experiments, then I think that's a good time to start looking at superloading considerations. And then I'll pass it over to Xuan if he has any additional comments.
Absolutely. So for the first part of it, for any single cell experiment, because the scope you are looking at is pretty limited, you only have like, say, a maximum of 100K cells. So the first thing you need to do is to know what is your rarest cell population in there, and whether this number can reach a decent amount of those type of cells in there. For example, hematopoietic stem cells, only 0.1% or whatever in the system. Can you get 100 good quality cells that give you a nice profile either at pseudobox level or at single cell level in this data set? If you cannot, you have to find a way to either flow sort or use some like max by the Miltenyi beads to enrich for those cell types, so you get a better thing. It's not like loading all the bone marrow in the channel and you can find hematopoietic stem cells. It's not possible. Yeah, and for the second part about like superloading and overloading, so probably if you look at the whitelist of cell barcode in 10x, you can know that the actual available jams, you know, all those jam beads, those gel beads are actually in the scale of millions. So the only reason 10x or other people load a small amount of cells is to avoid doublet or multilate. So like most of your cells would be in there, would be like singlet. The actual usage in a typical 10x flow experiment is only like 1 to 2% of the total gems. So as long as you could afford the multiple doublets or multilates, you have a way to tell them apart. For example, with hashtag antibody, with the hashtag oligos, it's totally fine to load higher. In our case, we load 130,000 cells. That was using the HT kit, so it's completely different fluidic design and everything. But generally, I think for a regular X plus, I don't know, Gem X or regular 3', you can load at least 3 times more than what is recommended if you have a hashtag oligo. Yeah.
And since it pertains to the question, we'll do one more, and that was specifically what was the doublet rate from loading at 100K, and did cell hashing help remove those doublets?
Yeah,
resolve those doublets. Sorry.
Yeah, so for according to we actually did everything according to 10x recommendation. So for the HT, we loaded 130K cells. The actual recover is about half. So in a normal 10x experiment, you load 16,000 cells and you get 10,000 cells back. So the more cells you load, you will see a decrease in the recovery rate in terms of singlet. And the hashtag antibody in this case will definitely tell you what are doublet or multilate. For example, you have one cell that got stained 50% of the the hashtag UMI over there is hashtag 1. The other 50 is like hashtag 2. Then you know it is very much likely a doublet. And for us, we want to be safe than sorry. So we are very stringent in our cutoff for all these doublets and everything. We just want very high quality single cells, especially in the progenitor stage, because you have cells that express markers from, you could have progenitors that express marker gene from 2 completely different lineage. It's not like a peripheral blood. You can separate these T cell and B cell so easily just by like 1 or 2 markers. So be stringent or be loose and let it fit according to your application. And I'm happy to answer any question, either just find me on LinkedIn or whatever email if time is running short. Yeah.
So thanks again, Xuan. That is all the time we have today. Amazing presentation. Thank you for staying on for some additional Q&A, and also to Sam and Roman, and of course everybody in the audience for joining us and contributing to this robust discussion. The recording should be up on the 10x website within a week or so. Thanks again, everybody.
Bye. Thank you.
Bye, everyone.